Hey guys, I want to go over chapter 21, the study guide, some of the terms. This is the first chapter in our last unit, which is evolution and Earth's history. So a lot of this unit is looking at how we're related to other organisms and how we have similarities and differences from them. And so we'll get right into this here. The first question I always like to start off with is which organism is more similar to humans or most similar of the three? So a fungus, a bacteria, or a plant. Let's take a moment to kind of think about that. Which of those three is most like us? And you can pause this if you need some time. So the correct answer is that animals, us, are most closely related to fungus. And that might seem a little bit um, difficult to understand. Most people think animals and plants are most closely related to each other. But fungus and animals both actually get their energy from um, taking it from another organism. So fungus grow on things and they basically leach energy from them, whether it's something that's dead or something that's living. And of course, animals, we eat other things. Plants take their energy from the sun, of course. So then animals, fungus, and plants are here. And then we have archaea and bacteria, which are both bacteria types. Bacteria here is like the common stuff you get. So you get strep throat. You have bacteria in your intestines to help you digest food. Bacteria that's in like raw meat. All of those things belong in this category here. The archaea bacteria is bacteria that grows in a really extreme environment. So this might be in the depths of the ocean. This might be in a volcano or the glaciers in the Arctic Circle. So very extreme conditions. Now you'll notice here that there's sort of a branching tree, and the branching tree shows how closely related we are to other organisms. So if you notice down here, mammals are most closely related to birds and reptiles, more so than we are to fish and insects. And as we move further and further away, we get less and less relationship until we get all the way down here to the other end where all the bacteria is. So a lot of this unit will be uh, how does this tree look and what does that tell us about how we're related to other organisms? So one of the ways that we study that is through genomics and proteomics. These are two terms on your study guide. Basically, the difference is we've done most everything through genomics, which has been like the genes that we've been looking at. So we took our samples of DNA and we cut those and we looked at them in the gel and saw what they were being expressed as. So the proteomics part is what the protein is that's being made. So proteomics studies proteins, genomics studies DNA or RNA, and how. So these two things don't always correlate exactly, and we've talked a little bit about that. There can be mutations. Sometimes those mutations change the protein. Sometimes they don't. So studying both of these together can give us two different views of how closely related we are to other organisms. That leads into the Human Genome Project. This was sort of the launch into genomics, which is the study of the DNA. And this was a long project from 1998 to 2003. Now we have 3 billion letters in our DNA, in our genome. So the genome is, of course, all of our DNA letters, and roughly 20,000 genes. So that means roughly 20,000 proteins, but sometimes that makes more. Sometimes a gene makes more than one protein, so that gets a little bit more complicated. Many of these are unknown. We don't know all of our genes. We don't know where they're all located. That's what this project sort of launched, and what the genomics is. The 3 billion letters was what the Genome Project sort of outlined for us. And so that's been really valuable because of all of the things that we've learned from it. And I've shown you guys like um, my 23andMe with a lot of the results from that. That's just one of the things that have come from it, but then also, of course, understanding of disease and cancers and all that has been very helpful. So how they did this is they use what's called a DNA sequencing method. And basically what this is the first time they did this, the DNA, and this is a gel like we've used in the past, so gel electrophoresis, where the pieces of DNA move along this length. And the ones at the top here, like the A, that would be the longest piece of DNA. And as you move further and further down, then those are the shorter pieces because they can travel further. And so when these are cut at different lengths, that gives us the sequence of the DNA. So if the shortest piece is a C, you know that that's the first letter because that's the shortest piece of DNA. The next shortest is an A. The next shortest is a G. And they would go through and through and through and all this, 3 billion letters long. That's why it took such a long, long period of time for them to actually map this out the first time. So they'd actually had to go through letter by letter to get this sequenced out. What are the letters of that genome? Now, today, we have a lot of different methods. Here's one. These are much faster. So this method basically has taken, you can ignore everything else here except for the letter C. And C represents blue. A is represented by green, G is represented by black, and T is represented by red. And so basically a similar thing happens where each piece of DNA is tagged with this fluorescent color, and then it's read out on a machine. But since the machine can detect colors much more rapidly, rather than a person going through and writing down, oh, this is an A, then they write down a C, the machine can read this out much more quickly. And so as a result of that, these new sequencing methods go from like a $3 billion project down to about 5 cents per 1 million bases. So it's much more in cost efficiency. So like I said, I think my genome to get all the genes that they had was like $150. And that was quite a few genes. So that's much cheaper than this $3 billion genome project. And so then the last thing here is one of the things that we look at is not just DNA and proteins and things like that, but we also look at fossils. So fossil records tell us a lot about how old things are, and we'll get into that a little bit more. But we use this thing that's called a geologic column. And a geologic column basically has the history of Earth outlined in it. Now, it's kind of a puzzle because each piece is fragmented. So if you look here on the map, in Canada, a little bit north of where we are, layers 17, 16, and 15 fit there nicely in a column. Now, things like wind and erosion and other things can kind of disrupt some of those layers. So if you look further south of this one, this is probably South Dakota-ish or somewhere in that area, columns 1, 2, 3, and 4. And you notice some of the columns are thicker or thinner in some cases. But then we start piecing these all together across the Earth, and we start to get an entire column from it. So 1, 2, 3, and 4 is here. 3 and 4 is really important because they match up with this column here. That allows us to do 5, 6, and 7. And so piecing this all together, like I said, like a puzzle, gives us this sort of sequence. And then from that, we can tell what fossils, for example, are in layer 11 and compare them to layer number 8. So which one is older? Which one is there earlier? Which one died earlier? Well, layers 1, 2 through 8 are much older or had died earlier than these layers here. So we can compare them to each other. We'll get into figuring out actually how old each of these layers are too as we move throughout this unit. But that's it for chapter 21. And if you have questions, as always, you can email. Otherwise, feel free to use your book. This study guide's pretty short, so that's it for today.